Good morning, everyone, and welcome to Soup and Science. Before we begin, we would like to acknowledge that Mcgill University is on land which long served as a site of meeting and exchange amongst Indigenous peoples, including the Anishinaabe and the Haudenosaunee nations. We acknowledge and thank the diverse Indigenous people whose footsteps have marked this territory on which peoples of the world now gather. I'm going to pass the microphone over to Professor John Sticks, our Associate Dean of Research.
Thank you very much, Pretty, and really great to see everybody here today. 51 attendees, isn't that great? It's so excellent. So I'd like to welcome everybody to the 33rd edition of Soup and Science, and we're joined today by four wonderful speakers who are going to tell us about their research. Now, after their presentations, you, the students, will have an opportunity to answer a skill testing question about each presentation. And the top 10 students who answer the questions correctly in the fastest time will win a prize. And that prize is a $25 gift card to the Lou James bookstore. So we look forward to your participation in that. And after this skill testing question event, we will have a moderated question and answer session, which will occur in breakout rooms. And there will be one speaker and one moderator in each breakout room. And so you'll be able to choose which room or rooms you want to visit and join the breakout room on your own. And then a few minutes before noon, before we finish, we'll return to the main session for a few final remarks. Okay, so I'd like to introduce our first speaker today, Professor Huy Bui, in the Department of Anatomy and Cell Biology. And Can does some very interesting things regarding the structure of macromolecular assemblies. He's interested in cryo-electron microscopy, which interests me too. And he is also interested in mass spectrometry, which I am also interested in. So I'd like to welcome you, Han, and it's over to you.
Thank you, John. I still haven't been able to share my screen yet. Yes, it's okay now. Okay. So good. So it's my pleasure to talk in this wonderful Soup and Science event. To introduce myself, I'm a structural cell biologist, meaning I'm studying cellular structure to understand its function for a living. Right. So to study structures, I'm using cryo-electron microscopy or cryo-EM. So what is cryo-EM, right? So to do cryo-EM, it's a technique that you first have to purify the protein. Then you have to transfer it into the EM grid. And then you have to freeze the sample very quickly in liquid ethane and liquid nitrogen temperatures. This allows the protein to really trap in a very thin vitrified water or ice layer without using any fixated. Because the sample is fully frozen hydrated without any fixated, it really preserves the structures and the resolution of the protein for later imaging. So after that, we have to bring the EM grid into the electron microscope and image it. We collect thousands and thousands of images in the microscope in liquid nitrogen temperatures, right? So if we get all the image, how do we get the 3D structures, right? So each of these micrograph here look like, really garbage, but it's actually, it contains each of these is a picture of the protein complex in many different orientation. So here I just highlight some of the orientation of the protein in here, highlight in a box, right? So if we able to pick out all the protein in like all the micrographs that we have, we can computationally figure out the orientation and back project it to get a 3D structures, just like you go to the hospital doing a CAT scan, right? So for example, here, I'm showing here, this is the structures of cryo-EM structures of the SARS-CoV-2 vaccine candidate, a full-length spike that these structures allow people to look at the conformation of the spike compared to different variants and also see if it's suitable to use as a vaccine as well, right? So in our lab, we are not working in COVID sample. We're actually interested in the cilia. So we're using cryo-EM to study the structure of the eukaryotic cilia. So cilia are like hair-like protrusions, such as like the term of the sperm, the tail of the sperm that allow it to move, or like in the Greek agar for it to swim. Or like in the respiratory cilia, where it act like a brush to like clear out the mucus and dust in your respiratory tract. Because the cilia is so important. The effect in cilia actually create, like a wide range of disease, from respiratory disease to low motility sperm and many others. But since the cilia is very conserved from very simple organism to human, we can actually study the function, the structure of the cilia in simple organism like this green algae, like to understand the function in human, right? So for example, this is a structure of the cilia from the chlamydomonas here. And if we get the high resolution structure using cryo-EM, we would have it look like this. And in here, I just show that it's really complex, which each color represents one protein. So in our lab, basically, we're interested in study the function of each protein like this, using molecular genetic, biochemistry, and structures to hopefully contribute to the understanding the cilia function in human and human disease. And yeah, so that's it for me. And this is my lab pictures taken before the pandemic. Thank you.
Thank you very much, Ken. And I look forward to future pictures like that last picture, and when things get better. So thank you very much. That was super interesting. And our next speaker is Professor Anmar Khadra of the Department of Physiology. Now, Anmar is interested in mathematical and computational approaches to immunology and endocrinology. He's interested in stability and nonlinear dynamics in these systems. And he's also a very active member of CAMBAM, which is the Center for Applied Mathematics in Bioscience and Medicine. So thank you very much for doing this, Anmar, and over to you. Over to you.
Anmar, you're on mute.
Sorry about that. I have to start this all over again. Okay, thanks for the introduction. And as I was introduced, my work is mostly focused on computational and mathematical modeling of biological systems. And typically, we use quantitative methods to conduct this research. And this is typically done using two different avenues. One of them is developing mathematical models or biophysical models. These are typical examples that you see here. They could be either differential equations, ordinary differential equations, or partial differential equations. Or we could develop computational techniques or algorithms that analyze the experimental data that we have, or even fit this experimental data to the mathematical models that we develop. So this is basically the general framework of the methods that we use in order to study these biological or physiological systems quantitatively. Now, what type of biological system we tackle? Actually, we don't have anything specific. We could analyze immunological systems, immunophysiological systems, or even cell biological systems. So here I'm going to give you some examples of the projects that we pursue in the lab. So one example is mostly focused on single cell electrophysiology. So here I'm showing you just the fluctuations in membrane voltage that you see in one part is called the GnRH neurons. So we take this electrophysiological data. We develop mathematical models, ion channel based mathematical models that mimic that type of behavior. And that would allow us to basically explain how these ion channels interact together at that type of activity that you see here. This is, for example, at the single cell level. And then we try to understand the downstream effect of this on hormone release, how this hormone is generated as in a pulsatile and episodic manner, how these ion channels produce that type of activity at the hormonal level. So these are rhythms that are detected in experimental data And we'll try to reproduce these rhythms and try to explain how they're generated. But these rhythms could be also produced at the neural network level. So for example, this is area V4 in the individual cortex, where you see at one point when the monkey is making a saccade in the eye, when making an eye movement, suddenly you see at time 0 exactly here, a traveling wave that goes to our region that is produced by many neurons that being recorded simultaneously using multi-electrode recording. So we wanted to understand exactly how this is generated. So what we do is that we, aside from using machine learning to explain how these activities are generated, we also develop mathematical models that are neural network models that mimics the topology of the neural network that is manifested in the core of the visual cortex. And we try to basically play around with different parameters of the system. For example, the coupling strength of the network between different neurons, and also the magnitude of the inhibition that is exerted within that neural network. Of course, we could manipulate other parameters within the model. And we try to see how this affects the dynamics of the system. So we could see here, for example, in certain parameter regimes, we could have synchrony. Other parameter regimes, you could have traveling waves, for example, that is in the red box, or traveling bumps that you see here. All right. And then we use machine learning again to basically see how the different activities that are produced by this network without actually manually detecting this by ourselves. So we could basically say in this parameter regime, we have synchrony, in another parameter regime, we have traveling bumps, etc., slow traveling waves, fast traveling waves, etc. And we do this over many different parameters and look at different parameter and see what the underlying dynamics are. And then we could basically study how behavior changes going from one to another. And that would give us some insights about what's happening, at least at the neural network level in vivo. Okay, so I'm giving you 2 different examples, but typically when we do all this work, we try to accomplish all of these things that we see here. We do all of these computational, this type of computational work to make predictions, to validate mechanisms, generate hypotheses, test hypotheses. Of course, also generate technological tools that could allow us to manipulate these systems at one point. And that's exactly my whole talk. So thanks for listening.
Thanks very much, Anmar. That's really interesting work. We really appreciate it. Our next speaker is Laura Latendresse, who is a student in environmental sciences. And Laura will be talking to us today about energy and Indigenous communities. Over to you, Laura.
Thank you. So hi, everybody. My name is Laura Latendresse, and I'm a student in the Baylor School of Environment with a minor in geography. And in the summer of 2021, I received a social equity science undergraduate research award, and that's what allowed me to do the research that I did last summer. So just a little bit about my project. I studied energy poverty, which is the situation of a household unable to access or afford adequate energy services to meet like basic household needs like cooking, cleaning, or heating. And it's a topic that hasn't been studied extensively in Canada, and specifically in Indigenous communities. We focused on Inuit Nunangat, which covers most of Northern Canada. And it's a topic that's related to broader issues, including health, environmental justice, and discussions over renewable energy implementation. So I think it's a topic that deserves a lot more scrutiny, but basically I looked at gray and scientific literature as well as data from the 2018 Canadian Housing Survey to get an idea of the challenges that these communities face and how they relate to energy poverty. And we found that really there are unique challenges and barriers facing these communities that contribute to concerns over energy poverty. And we focus namely on housing, and how inadequate homes with poor ventilation and insulation can contribute to higher energy bills and uncomfortable living conditions that contribute to health concerns as well. Now, more broadly, my research taught me a lot as a student, as a researcher, and about what I want to do in the future. I learned about how to stay organized, about how to persevere in difficult times. I learned about how to do research, communicate with my supervisor, meet deadlines, all that good stuff. So it was really valuable for me to learn about this project, to kind of delve into a topic that I knew very little about. And yeah, that's it. So I know that this can be really scary for a lot of people, and I'm more than happy to talk further with you guys about just what it means to do research as a student. And yeah, that's all. That's it.
Great, Laura. Thank you very much. It's really a super interesting topic. So let's move to our last speaker, Toni Spilfogel in the Department of Chemistry. And Toni's going to be talking to us about halogen bonds, which I like halogens. So I'm very interested to hear your talk, Toni.
Hi, everyone. Can you see my screen?
Yes, all good.
Okay, perfect. Okay, so yes, I'm going to be talking a little bit about halogen bonding and my research, but also an approach to research that some of you may not have heard of before, and a way that you guys can all get involved with research at McGill. So my research journey at McGill started in January of 2020 when I reached out to Professor Thomas Law-Frischig to get a research position in his research group for the upcoming summer. And pretty quickly, we realized that that was not going to be able to happen unless we could figure out a way to do that online. And that's when Dr. Hatem Titi recommended that we do a database mining project where we look at the Cambridge Structural Database, which houses all of the new structures that are published every single year. So we wanted to look specifically for halogen bonds, which is a type of non-covalent interaction, very similar to a hydrogen bond. And we wanted to look for these halogen bonds in porphyrin systems. So porphyrins is this structure right here. It may seem unfamiliar to a lot of you, but you're actually more familiar with this molecule than you think, as it's what makes blood red in heme, and it's what keeps plants green. It's in chlorophyll. So basically, we wanted to look for these halogen bonding interactions in porphyrin systems like here. And as you can see here, we have this double pie chart, which actually shows the distribution of all of these halogen-based interactions that I was able to find in the CSC. We found 144 unique structures that had different types of halogen bonding interactions. And we're actually able to take this a step further, as when we looked through our data set, we noticed that there was these architectures that we kept on seeing over and over again. So we're actually able to classify all of the porphyrin halogen bonding interactions that we saw based on one of these 13 different types of motifs that we saw. And we were really excited about this. So we were actually able to publish it less than a year later after starting the project. I had gotten my first author research paper, which is something I'm very, very grateful for. So now I'm just going to quickly shift gears and talk about how you all can get involved with research at McGill. I'm sure a lot of you have heard of some of the research awards like the SURA and the NSERC, and I highly recommend applying for these different grants, but the thing is that they're awards, and obviously not everyone will be able to win. But what everyone can do is take a research class. Now, the research that I showed you all actually was the research I did for my Chem 396 research course. Now, if you're interested in research at McGill, I highly recommend doing a 396. Every single department within the Faculty of Science offers a 396. You don't need to be an upper year student to do it, and you can even take it with a professor outside of your major. Now, once you've done a 396, which is just an amazing way to get acquainted with research at McGill, there are many other department specific research classes you can take, such as honors projects and independent studies. I've personally done 13 research credits here at McGill, and I highly, highly, highly recommend looking into that if that's something that you're interested. You can visit the link that I've included on my slide. Feel free to take a screenshot of it, or you can also visit your department's website and they should have those specific courses as well. So, you know, I just really want to stress the fact that don't let the pandemic, the situation that we're currently in, stop you from trying to get a research position. And not all research needs to take place in a lab. I think that we've kind of seen that really echoed today. And really check out this link that I've posted. It's a really great way to get involved with research here at McGill. So thank you all so much for listening.
Wow, Toni, thank you so much. That was a great talk, and what a nice way to end the 4 presentations today in terms of showing the students here how to get involved in research. So I totally concur with what you said. If you're interested at all in research, you folks out there, reach out starting now, and reach out to some professor that interests you, whose research interests you, and see what they might have going for you. Do not hesitate. Really, really great.